We are from Group 7, and we all know that China is well known for its tea and the rest of the world, country in the world. And this morning we'll be talking about biological control in terrace and forest tea plantation in Mongson. Okay, before that, we all know that pest control usually comes along with the term with synthetic pesticides, but we know that a lot of international organizations have established a low and stringent maximum residue limits on crops, and it's not a good way to apply pesticide on crops. So we'll be talking a little bit on biological control. And what is biological control? It's an environmentally sound and effective means of reducing or mitigating pests and pest effects through the use of natural enemies. And conservation and application of natural occurring biocontrol agents are more preferred than introduction of exotic predators and parasitoids for the better efficacy and to avoid ecological problems. We'll move on to our project site. A little bit introduction of Mongson. And the Mongson Tea Mountain is located around 31 kilometers from Monglang town, and it has an elevation of 1500 to 1800 meters, and it produces around 120 tons of high quality tea per year. And there's two types of tea plantation in Mongson, the terrace tea plantation and the forest tea plantation. And we have asked around the farmers around the tea plantation, and most of them doesn't use pesticide, but herbicides are only practiced once a year. And when you have a condition as this, where pesticides is not introduced, that's where you will get a lot of phagocytous herbivorous pests. And you need a good biological control agent to control the pests. So these are pictures of the terrace tea plantation and forest tea plantation. Okay. Our research question: does forest tea provide better biological control than terrace tea? So how does this experiment is done? So we will look into the abundance of pests, the abundance of predator, and we will also look into the leaf damage percentage, and as well the habitat complexity factors. The habitat complexity factors here are including the tea tree density, the number of large trees around the tea plantation, basal area, percentage of understory coverage, and as well as distance to the nearest forest. Next part we will talk about our methods.
I will continue to explain the method of our experiment. First, we look at our study area. We choose the 5 terrace tea and 4 forest tea. The red dot is refer to the terrace tea, and the green one refer to the forest tea. This is an example of our terrace tea plantation. In each plantation, we set up 2 plots of 10 by 15 meters. In each plot, we placed a transect line from north to south, and the two plots were placed at a relative distance from each other. At each plot, we counted the number of big trees, calculated the basal area, and we used 3 quarters of 1 by 1 meter to estimate the understory coverage. From this picture, you can see this is a plot and this is a transect. In each plot, we put a malaise trap to capture the insect. We use the point center quarter method to select our sampling trees. First, for each transect, we mark 5 points, and at each point there are 4 90-degree quadrants. At each quadrant, we choose the nearest tree as our sampling tree. So in each plot, we have 20 tea trees. We also measure the distance from the central point to the nearest tea tree in order to measure our tea tree density. For insect collection, we use mass trap to capture the flying insect. And for our sampling tree, we collect insects by hand, and aspirator we use to collect the smaller insects. For the leaf damage measurement, we choose one branch from the middle portion of each sampling tree, and five leaves were scanned of each branch to estimate the percentage of leaf damage. Okay, for the data analysis, we use char chart to show the shared species in both tea plantation, and linear mixed expect model was used to compare the pest and predator abundance, percentage of leaf damage between forest and terrace tea. Lastly, we use CCA to analyze the influence of habitat complexity factors on insect composition. And next part is our girl. For our result, here are some of the pest insects that we found in the forest and terrace tea, and most of them are mostly Lepidopteran larvae and tea aphids. And here are some predators that we found in the terrace and forest tea, and the dominant predator mostly are spiders and also these overfly larvae. This is our results from the Malay trap, and here we found that the abundance and the species richness of parasitoid and pest insects in the forest are lower than in the terrace. The forest Malay trap captured lower insects, and our analysis is probably because in terrace there are lower wind speed compared to in the forest there are lower wind speed compared to the terrace tea. And here are we found that there are 5 shared species of parasitoids and 2 shared species of pests in terrace and forest tea, and our Chow-Jaggard estimation suggested that the forest and the terrace has similar diversity. This is the result of the insect data that we collect from the hand collecting method, and we found a similar result in which that the forest tea has a lower abundance and species richness compared to the terrace tea. And for this shared species observed, we also found that the predator in the terrace, there are 31 shared species of predator in terrace and forest tea, and 37 shared species of pest. And the Chow-Jaggard estimation also suggested that there are similar diversity on both terrace and forest plantations. We use this is the relations of the abundance of pest and predator between the forest and terrace tea. We use the log transformations for the pest and predator abundance, and we find that there is no significant difference in the pest abundance between the forest and the terrace. And we also found a similar result in which there is no significant difference with in a predator abundance between the forest and the terrace. For the leaf damage analysis, we use the arc-sine square root transformations for the percentage of leaf damage. And in this figure, we can see that we also find a similar result in which there is no significant difference in the leaf damage percentage between the forest and the terrace. We use the CCA ordinations to see the relationship between the habitat complexity factors with the past species composition and further species compositions. And in this graph, based on the CCA analysis for the past composition, we found that the CCA axis explained 29.5% of the variance, and CCA2 axis explained 27.4% of the variance. And we found that T density and also basal area are the most the habitat factors that mostly explains the species composition and distributions of pests in the terrace and forestry. And in the for the predator, the CCA1 axis 1 explains 35.7%, and CCA axis 2 explains 26.8%. And we also find that the T density and also the basal area are the two habitat factors that mostly explain the species predator species composition in terrace and forestry. So we go back to our previous questions. Does forestry provide better biological control than terracity? And based on our results, we found that forest and terracity has a similar biological control, And from our analysis,